Okay, welcome everybody. Today we're going to be talking about the Data Viewer Integration Messaging Protocol. So let's go here. In this webinar, we'll start by just giving a very brief overview of Data Viewer. If you'd like more information about that, you can check out our Data Viewer video tutorials at our support webpage. Let me just exit out of this quickly and show you what I mean by that. So I have our support forums opened here, and I've already navigated to our how-tos video tutorials page and then gone to the Data Viewer video tutorial series. And you can see here it's a 10-part video series. Each video is maybe 5 to 10 minutes. So the whole thing is maybe about an hour long. And it covers the basics of Data Viewer. So if you're not familiar with that at all, then you can check out this video series to become more familiar with it. Let's go back here. After we give a brief overview of Data Viewer, then we'll talk about a brief overview of the EyeLink API, which is what allows experimental programming environments like Psychtoolbox, PsychoPy, EyeLink Presentation, Python, C, C++, C#, etc., to communicate with the EyeLink systems. Then we'll jump into the main topic of the webinar, which is how you can use the Data Viewer messaging protocol through the EyeLink API to make sure that the data files that you create are formatted in a way that makes them friendly with the Data Viewer software. Okay, so first, Data Viewer is an analysis package for eye tracking data, for EyeLink eye tracking data, and it allows you to visualize the data in different ways, apply some filters, and do some processing to the data, and also analyze that data. So export summary reports that kind of give you information about where the participant looked during the course of the experiment. It runs on Windows, Mac, and Linux operating systems, so it's pretty flexible. And let me just show you quickly some example visualizations here. So on the left here, we have what's called a temporal graph view, which can show the eye position over time. So on this axis, we have like the time is going along the x-axis here. And then along the y-axis, we have position. And this graph is showing the position of the eye, which is the red and green lines here, along with the position of a moving target in a smooth pursuit study. So these black and blue lines here show the position of the moving target over time. And so you can see, you can kind of easily compare the position of the eye over time to the target over time. On the right here, we have heat map. So it's kind of showing through these colors here where the participant was tended to look during that trial of the experiment. And you can create heat maps, fixation maps, as we call them, pretty easily through the Data Viewer software, and it has some other visualization options as well. We're going to be jumping back and forth to Data Viewer later, so this is just a quick overview. Here's another view, which is called the spatial overlay view. And in this, you can see these circles represent eye fixations, and these kind of orange boxes and ellipses and freehand shapes are interest areas that have been created to kind of highlight parts of the stimulus that are of interest. And in using Data Viewer, you can pretty easily match the gaze data to these interest areas and then output tab delimited text files that we call reports. And those reports can give you summaries of the data that can then be put into analysis packages. So when you create a report, you get a little dialogue that looks something like this. And there are lots of different reports. You can create fixation reports, saccade report, interest area report, sample report. And basically these different reports are formatted in different ways, and the name of the report tells you what each row of the output file will be. So here it's creating a fixation report, which means the output file will be formatted such that each row of the output file is a different fixation. And when you create the report, you'll see a dialog like this where you can specify what the columns of the report will be. And so you can pretty easily create files that can be exported from Data Viewer and then imported into statistical packages like R, Matlab, SPSS, those types of things. So Data Viewer is kind of like an intermediate step between data collection and statistics that does a lot of kind of the grunt work of like matching the gaze data to the fixations and then generating these reports for you. Okay, so that's a brief overview of Data Viewer. Now let's talk briefly about the EyeLink API. Maybe I should mention before we jump into that, if you want more information about a particular usage of a programming, like how to use the EyeLink API in a particular programming environment, let me jump back to our support forums here. If we go to the main support forums and then go down to this getting started, we can go to getting started with experimental programming, and we have all these different getting started with like PsychoPy, presentation, etc. And those can help you to become familiar with the specifics of a particular programming environment. So I encourage you to check those out if you're not familiar with using the API in general. But let's go back here. In a typical like EyeLink setup, you have two computers involved. So you have one computer which we call the display PC, and it's the computer that's running those experimental applications, PsychoPy, Psychtoolbox, presentation, ePrime, whatever. And it's presenting the stimuli to the participant. So here we have an old CRT. They have great timing properties, so don't judge there. But basically, the participant is looking at the display PC monitor. That's where the experiment's being run. And the experimental application is using the EyeLink API to communicate with the host PC. And this communication is done via an Ethernet connection between the two computers. So basically the display PC talks to the host PC and gives it kind of commands that allow it to do certain things. So down here, I've kind of highlighted some of the important API functions. This is by no means an exhaustive list, so you can kind of check out documentation of the API, which you can get from our support site as well if you have more questions. But basically, it will allow you to perform certain eye tracker functions like calibration of the eye tracker, validation, drift check. It allows you to tell the eye tracker when to start and stop recording eye data. You can send commands to change settings on the host PC, and on the eye tracker. So any of the host PC parameters or settings can be configured via a command from the display PC to the host PC. Importantly, you can also send messages from the display PC to the host PC to mark the occurrence of experimental events. So like when you're running an experiment, you're going to have all these different events that are going to be happening on the display PC. You know, you're going to be presenting different trials with stimuli here. And you want to send markers to the data file that log the time when these different experimental events occur. And that's what this is referring to. We call them messages. That's our little lingo. We call these markers messages. And so you're going to send messages to the host PC to mark the times of these experimental events. But importantly, and this is the topic of our discussion today, you can also send messages from the display PC to the host PC to communicate critical experimental information to the Data Viewer software. So you send specially formatted messages that when Data Viewer imports the EDF file, that's the EyeLink data file, when Data Viewer imports that EDF file that has these special messages, it interprets them in a certain way and allows certain features to be used in Data Viewer. And so that's what we're going to talk about today. Okay, so here's a I'm not even sure if this is totally exhaustive list of everything that you can use, but this has a lot of the different programming environments that can access and use the EyeLink API. So you can see they're quite a quite a lot of options here. And again, you can get more information about these on our website. Okay, so the messaging protocol. This is the big topic of today. As I mentioned, when you send these messages, it's often used to mark experimental events, but you can send these other special data viewer messages. And these allow you to create EDF files that are compatible with the data viewer software. And we're going to go through each of these in detail, but I kind of listed the different ones we're going to talk about here. You can kind of refer back to this slide later to just kind of remind yourself of all the different options. But we'll kind of just go through these one by one here. So basically I'm trying to, in this slide, describe the type of message here and then give a quick little example of what that message looks like, okay? And so these are the different things we'll talk about. Let me exit out of this for a second and just show you an example. I have a Matlab script opened here, and I just wanted to show you what this looks like in context of an experimental programming package. So here you can see this is an API function call. And basically we're saying, okay, EyeLink system, let's send a message to you. And this is the text of the message. So up here we have an event mark. Up here we draw the trial stimulus to the screen, and as soon as we draw it, we send an event marking message here to just say stimulus onset. And then right after that, we send some special data viewer messages. This one, just for an example purpose, is used to allow data viewer to like load the trial stimulus so that you can see it in the background of data viewer. So see how we can see the trial stimulus here. That's the result of one of these image load messages. So let's go back here. So basically we're sending an image loading message, then we're marking some interest area on the screen. Okay, so let's dive into these in a little more detail now. And let's just start at the beginning. So at the very beginning of your experimental script, you want to convey to data viewer the resolution of your display PC monitor. So by display PC, it's the computer that's presenting the stimuli to the participant. And so you can see here, you would send a special message to the EDF file that is formatted like this. It says display coords, and then it has the top and left and top, top, right and bottom coordinates of the monitor based on the monitor's resolution. And so this will be sent at the beginning of the experiment before any of the trials are run. And really what this is doing is allowing data viewer to format this window here. So it's going to format this window based on that resolution that you specified. This is called the trial view window. And so it's critical to send that type of message at the beginning of the experiment. Oops. Okay, and this message should be 0, 0, which is the left top, and then it should be the width -1 and the height -1. Since it includes 0, we need to subtract 1 from the overall resolution so that we have exactly 1920 by 1080 or whatever resolution you're using. And all our examples in the different programming environments show how to do this and show how to do most of these other messages too. Okay, so let's move on to the next one. When you are running an EyeLink experiment, you're gonna typically be saving one EDF file or one EyeLink data file for each participant. But you want that data file to be broken down into multiple trials. And so at the beginning of each trial, you would send a message that marks the onset of that trial. And so the formatting is trial ID with no spaces or anything. And then you can just put the trial number afterwards. It looks like I missed a space there, but basically it should be trial ID space 1, trial ID space 2, etc. So each trial, you just use a larger number. Technically, this doesn't have to be a number, but it's kind of convenient to do it that way so that you can easily identify the trial number in the data file. Okay, and this should be sent at the beginning of the trial. It marks the onset of the trial, and it allows Data Viewer to break it into that single EDF file into multiple trials. And there's another message that's sent at the end of the trial, which we'll come back to, that just marks the end of the trial. And so when you load an EDF file or import an EDF file into Data Viewer, you'll be able to see, like, for example, see how this is the EDF file name here, and see how Data Viewer has broken it down into the different trials. That's a result of these trial ID messages. I don't know why it's kind of freezing up here. Okay, there. So that's what we do at the very beginning, mark the beginning of the trial. Then as our different experimental events occur, we are gonna, of course, send like trial event messages. And these aren't really technically like Data Viewer integration messages, but you're gonna need them in Data Viewer. And so like throughout a given trial in your experiment, you're gonna be recording eye movement data continuously. And you want to mark the time when these different experimental events occur. So I'm just imagining here like an experiment where you have an image, a question, a button press, and then some feedback. And you want to mark the times of these events in the data file so that when you go to do your data analysis, you can focus in on certain parts of the trial. And you focus in on those certain parts of the trial by setting interest periods in Data Viewer. And you can check out our video tutorial series about how to do that. But basically, when you're doing analysis, let's take a quick look. We have this interest period specification box up here. And you can click edit here and bring up an interest period manager. And then you can specify like a message to start the interest period. And you can specify another message to in the interest period. And then once you set that, you'll only be looking at data from that part of the trial, and it'll apply to all the trials in your Data Viewer viewing session. So it'll apply to all these trials here. Okay, so basically we're going to use these messages to mark the times when these different events occur. And like one kind of rookie mistake that people make is sometimes they try to put like condition information or like independent variable values as the event marking messages. And that's not really what we want to do, because then when you set the interest period, it would only apply to the trials that match that particular condition information. And usually instead, we want to have a consistent messaging text across trials so that when we set the interest period in Data Viewer, it applies to all the different trials in the viewing session. So that's kind of an important thing to remember, is just be consistent in your messages for the most part. There's a few kind of exceptions where you might not want to do that, but generally you want to be consistent. Okay. The next type of message is called an image load message. And this is a critical message. This is what allows you to see the experimental stimuli in Data Viewer as a background over which the gaze data will be overlaid. And so, like, just to jump back to our example, remember I pointed out that as soon as we draw the screen here in Matlab, we send an event marking message here, and then you can see this is one of these image load messages. And so this is where we're going to tell Data Viewer, okay, at this point in time, we want to load this image or whatever image it is as the background, and you'll be able to see that. And let me just show you. If this is a spatial overlay view in Data Viewer, but if I go to the trial animation playback view, let's stop it and play the trial. Oops, let's get out of it. Let's go to full trial period, sorry. Okay, so we can see so in this example, there wasn't actually a question. They saw a picture, then pressed a key to respond to it, and it gave some feedback. So there was one image load message that was being sent when the trial stimulus appeared, and then there was another image load message sent when the feedback appeared. And that's what enables us in the playback to see the kind of trial experimental stimuli unfold as they did for the participant during the trial. Okay, so you can send these, you're not limited to just sending one of these is what I'm trying to say. You can send these several times as the trial unfolds, and that way when you play back the trial in Data Viewer, you'll be able to see the images unfold as they did for the participant. Okay, and there are three types of these image load messages. So you can see here, this one's using image load center. And in that one, when you specify center here, then after you put a path to the image, and this path should be relative to the location of where the EDF file will be in your experimental folder after you've collected the data. But after you put that path, you can then put the XY position of where you want the center of the image to appear on the screen. So you can use a center coordinate scheme, or if you instead say top left here, then you would, instead of specifying where the center of the image would go, you would specify where the top left corner of the image will be positioned in the trial view window of Data Viewer. And then the third way is you can specify fill. So if you put fill here, then it will fit the image to the screen. So basically we'll either stretch or shrink it based on the difference between the image size and the size of the screen resolution. So there are a few different methods for how you can use this image load type of thing. I tend to mostly use the center one, but you can use any of them. Okay, the next type of message is a video frame display message. Okay, so this one can allow us to, it can allow Data Viewer to basically play back a video that was presented to the participant during the experiment. And what you do here is send a message that starts !V. A lot of these Data Viewer messages start with !V, by the way. Then you put V frame, then you put the frame number, and I've just kind of chosen a random number, 27 here, but you would use one of these messages for each of the frames of the video. Then you put where the top left corner of the video would go, and then you put the path to the video file. Okay, so let me just show you. I named it Frisbee here, and you'll see why in just a second. So let's exit out of here. And here's a video experiment, and I'm gonna go to the trial animation playback screen and see when we play it back, you can see the video playback. Ignore this guy here. That's me. That's my colleague Greg. But basically, this is able to, you're able to see the video here because of a series of these video frame messages that are sent to the data file. And just as an example, I have like a PsychoPy script here, and this is an example that shows how to do video playback in PsychoPy, and you can see it's basically got a loop set up here. And on each frame of the video, it's sending a different frame number, okay? All right, so let's go back to the, so that's what it will enable us to see the videos played back in Data Viewer. Okay, if you don't have image files for your trial stimuli, for your experimental stimuli, then another option is to send simple drawing messages. So there are a few of these options. This one clears the screen. You can draw lines, you can draw unfilled or filled boxes, and you can draw just kind of like circles with dots inside. That's called fixed point. And each of these types of messages requires different values afterwards. But just as an example, I have draw box here, and this is a RGB. This is the color value. So this will be kind of like a medium gray. And then you put the left, top, right, and bottom coordinates. And so this would draw a box with a top left corner of 300, 400, and a bottom right corner of 600, 700. So it'd be a box kind of like this on the screen. And that would show up in Data Viewer as well. So you can see the Data Viewer manual for more information about this. If you want to check that out, let me just show you how you can access that. If you open Data Viewer, you can go to Help, Contents, and if you go to this section, Protocol for EyeLink Data to Viewer Integration, you can see there are all these little you know, these are the messages we're talking about right now. And if you get a simple drawing command, you can see kind of the protocol for all these different drawing commands. And that goes for all these different things we're talking about. Okay. So you can do some simple drawing. And another thing you can do is like sometimes, like for example, in a video experiment, maybe you don't want to send a ton of messages to the EDF file. Maybe you, in trying to make your experimental code run more efficiently, maybe you would just kind of log all your messages to your computer's memory, and then at the end of the trial, dump all those messages to a text file. That's kind of a more efficient way to program things generally. You don't have to do that because really sending messages to the EDF doesn't take much processing power, but still it's a more efficient way to do it. So another way you can basically send drawing commands to the EDF is just at the onset of the experimental stimulus, the trial stimulus, you send one message, and this message can be called draw list, and it can point to a particular file that is going to exist at the time of analysis. It doesn't even have to exist when you're running the experiment. It just has to exist by the time you use Data Viewer. And then instead of sending like all the video frame messages directly to the EDF file, you can write them to that text file, and you can come up with whatever extension you want. Our experiment builder software uses this VCL. It does something like this, and it uses a VCL extension for these text files. But basically, you say, okay, here's a pointer to this text file, and then to that text file, you write all those image load messages. And when you write them to the text file, you don't need to put the exclamation V, but what you do need to do additionally is notice how all these messages begin with a number here. Those numbers are the time of the onset of those different stimulus characteristics. So you can see here at time 0, which is the time of the message. So basically the 0 point is the time of the message here. And at time 0, this will be probably the time where we initially like draw to the screen. We can clear the screen to some background color, and then we can draw our image at some certain coordinate. And Experiment Builder uses these weird image file names, but you could put whatever your stimulus file names are here, and then you use the position and size information here. And so you can see here at time 0, we draw this image, and then 5 seconds later, we clear the screen again and draw a different image. And then 1833 milliseconds later, we clear it again and draw a different image. So you can basically have all of your drawing commands in that text file, and then at time 0, at the time of the initial trial stimulus, you just send one kind of text file message. And so if there are a lot of drawing messages, this can be kind of useful. One thing I should point out, note these numbers are like, see how they have a negative value? Like negative means kind of in the future compared to the 0 point. If you had a positive value here, it would actually move it backwards in the past, and that's not usually what you want to do. You would usually have them kind of in order of presentation. And since they're offset values, which we'll come back to later, if they are happening after this initial event, then they need to have negative values. And the reason for that is because this value, this time value, gets subtracted from the timestamp of the initial message to determine the time when it occurs. So you can make sure to use negative values here. Okay, so that's a draw list message. Another like really kind of useful but specific to a certain type of experiment is a target position message. And a target position message is usually used in like a smooth pursuit study where you have some kind of moving target going around on the screen, and the participant's task is just to look at that moving target. And usually they're engaging in what's called smooth pursuit eye movements during this task. And so the idea is like, we're going to be recording eye movement data during the experiment, obviously with the eye tracker, but we also want to mark the location of this moving target in our data file. That way, when we go to do our analysis, we have an ongoing record of the position of this target on every screen retrace throughout the trial. And that way we can easily compare the eye position data to the position of the target over time. And this target position can be visualized in the temporal graph mode, and it can also be output alongside the gaze data when you create sample reports from Data Viewer. And that can include the target's velocity and acceleration. So it's useful, not just in the position, but also in getting the velocity and the acceleration of this target over time. And the sample reports can also include eye velocity, like they can include eye position, velocity, and acceleration as well. And so what you do here is you basically put !V target _ pause, and then the name of the target, and then the XY coordinate of the target at that particular point in time. And then here you can put a 1 if the target is visible, or 0 if the target is not visible. So sometimes people do experiments where the target like disappears and then reappears over time. And you can, when it disappears, put a 0 in your messages, and then when it reappears, start putting a 1 again. And that way, when you export the sample report, there's also a column that you can include that has the visibility status of the target, and that will help you with your analysis as well. And then this final little parameter here is whether the target should be considered to move in a stepwise manner, which is kind of in reality how it moves. Like on every screen retrace, it's not like just based on the way monitors work, it's not like moving smoothly all the time across the screen. It's actually like jumping from one position to another on each screen retrace. And so if you specify this value as a 0, it would operate in one way, and if you specify it as a 1, it will operate in another way. And you can check out the manual for a little more information of that. It's kind of a subtle difference, but basically, like, the eye tracker is going to be sampling data at a much faster rate than most monitors, well, really probably all monitors, can present stimuli. And so what this is doing is saying, like, between screen retraces, are we going to think about the target like moving in an interpolated fashion from one position to the next, or in a stepwise manner? Let me exit out of this and show you what so here's some smooth pursuit data that I have open. And right now I'm in this temporal graph view, and you can see in the temporal graph view, let me turn some of this stuff off. Like, this is time here, and this is position here. It's in screen pixel coordinates, and we can look at the X coordinate of the eye over time and the target X coordinate over time. So this black line you can see is like the target position over time, and then the red line is that the eye's X coordinate over time, and then this would be the eye's Y coordinate over time, and this is the target's Y coordinate over time. So these blue and black lines here, let me just show you, these lines here are only there because we sent the target position messages. And just to show you like what some of these messages look like in the data file, this is an ASC version of the data file. So as I've been mentioning that the data files are called EDF files. Those EDF files are binary, but using a converter that comes with the EyeLink developers kit that includes the API, you can convert the EDF files to these text readable ASC files, and they look kind of like this. So you can see they have a lot of information in them, but this is the point where we start recording data, and all of these are the eye position, are basically the eye data over time. And if we scroll down, you can see there's a target position message. There's another one. And so you can see at this point, it looks like it's still, see how the target position isn't changing, but notice here, now it's starting to change. So this is on every screen retrace. The software that was used to present the stimuli is writing these messages, and that's what enables us to see this position over time. Okay. Bring back the PowerPoint here. Then another thing you can do is send interest area messages. So these messages will make it so that when you import the EDF file into Data Viewer, these interest areas that you specify will be loaded automatically and just ready to use in Data Viewer. And you know, as is kind of obvious, when you're doing data analysis for an eye tracking study, you want to match the gaze data to these different parts of the screen, and interest areas are what allow you to do this. Some people call them areas of interest. We tend to call them interest areas, but same idea. So basically, when you program your experiment, you can send interest area messages, and this will allow you to already have those ready to go in Data Viewer. You don't have to do this. Let me point out down here, you don't have to create the interest areas through the messaging protocol. You can also draw them by hand in Data Viewer. And I kind of think of like, sometimes it's better to send them through messages to the EDF file, and sometimes it's better to draw them in Data Viewer. So as a couple examples, like let's imagine you were doing a visual search experiment where you have like 100 T's on the screen and 1 L, and in your experimental programming language you have already figured out, you know, where you're going to position all these T's and the 1 L. Like in that case, it's going to be a lot easier to send these messages than it is to draw those in Data Viewer, because you'd have to be drawing like, you know, over 100 like interest areas for each trial or whatever. So it's a lot easier to do that through messages in that case. For a different type of an experiment, like where each trial is just like a different image and you want to have interest areas for those different images, it's going to be harder to figure out the pixel coordinates of all these different interest areas of the image, and it's going to be easier to draw them in Data Viewer. So really, like, the decision of whether to do interest areas through messages to the EDF file or by drawing them in Data Viewer should be kind of motivated by the type of paradigm you're running. Let me exit out of here and I'll show you what I'm, let's go back to the spatial overlay view here and let's go to our image period. So you can see here's a rectangle interest area that could have been created from a message to the EDF file. And when you, if the EDF file had those interest area messages, when you load them in, then you would be able to see the interest areas already. You can also, as I mentioned, draw these interest areas. All right, that's kind of a stupid interest area, but you get the idea. You can also draw them after the fact. Okay, these interest area messages, like you can have 3 different types of interest areas: rectangle, freehand, and ellipse. And for rectangle and ellipse interest areas, you specify the left, top, right, and bottom, and then the label. So, or sorry, the ID, left, top, right, bottom, and label. So you can see here, we specify the shape here, and then we have the ID, the left pixel coordinate, the top, the right, and the bottom, and then the interest area label. Okay. Interest areas can also be dynamic, and if so, then you would specify the start and end time of each interest area instance. You can see the manual. There's a section that describes how to do this. Let me quickly show you. So if we open up the manual and we go to the interest area commands, this describes kind of the basics of interest area messaging format. And if you go down to the bottom of this, you can say, click on format of static and dynamic interest areas, and you can see more information about these are dynamic interest areas here. So you can see that there. If you have a lot of interest areas, or if you have dynamic interest areas that are changing position a lot, then you can do something kind of like the draw list thing, but for interest areas. So instead of writing the interest area messages directly to the EDF, you could alternatively just send an interest area file message to the EDF at the 0 point of, kind of like in the draw list, you send it at the time of the 0 point. And you point to a text file that would then in turn contain the interest area messages. And it's similar to the draw list, you know. So basically for static interest areas, the time of the onset should be specified at the beginning, just like in the draw list. So see how this is -200. That means this interest area starts, this is a static interest area, and it starts 200 milliseconds after the time of the interest area file message. And these are some examples of dynamic interest areas. And if you're going to create dynamic interest areas, then you need to specify the start. Like basically with dynamic interest areas, you have like, see how we have the same interest area ID and label. This is one monkey, and you can see that's used repeatedly. So we have monkey here, and we have two in giraffe. So we have giraffe here and here. And so these monkey, these are two instances of the same interest area, but this is a dynamic interest area, so it's moving over time. And you can see that from 200 milliseconds to 400 milliseconds, it is at this coordinate list. So this is the position of it from that for that period of time. And then later from 401 to 600 milliseconds, it's at this different location. And that's the way you can kind of make these interest areas move over time. Let me go back to our video example. So these are some dynamic interest areas. See how they're like changing position over time? That's what we mean by dynamic interest areas. Okay, so that's how you can do that. Let's go back. Okay, then another thing that you're going to want to log are the trial variables. So in your experiment, you're usually going to be manipulating like independent variables, or you're going to be like, changing trial conditions across trials. And so at the end of, usually at the end of the trial, like you would send these special messages called trial variable messages to log the different values of your trial variables. And these can be used for both independent and condition variables and for dependent behavioral variables like reaction time, accuracy, which key did they press, that type of thing. And so I just have a couple examples here. You can see we have one variable that's going to be called trial condition here, and it has a value condition 1. And then on a different trial, we might send a trial variable that says trial condition 2. And here we have a dependent variable called reaction time. And so the name of the variable will be reaction time, and the value will be, in this case, 2364 milliseconds, for example. And these are usually sent at the end of the trial. You don't have to, you can send them really whenever you want, but usually you aren't collecting things like, you don't know the reaction time or which key was pressed until the trial is finished. And so you would send these, at the very end of the trial before you send the trial offset message. And these allow you to group the data by the values of these variables in Data Viewer, but more importantly, they also can be included in any of the output reports that are created in Data Viewer. So let's exit out of here and let me show you what I mean here. So let's just bring up this image one. Okay, so by default, the data will be grouped by the EDF file name here. But if you go to Edit, Trial Grouping, then see we have all these trial variables that were sent in this experiment. So if we wanted to group by the image file, for example, we can move that over and regroup it. And now the data are grouped by these image files. And that way, if we wanted to like look at all the data for like a particular image file, we can do that up here. Okay. And critically, when you create any of the reports in Data Viewer, you're going to have the ability to choose from all these like predefined variables in Data Viewer that will become the columns in the output report. But if you scroll down, you can see you can also, they're always colored blue like this, but you can also include any of your own user-defined trial variables, and those would become columns in the output file. And usually you're going to need that condition or behavioral data in your output files as well. So that's kind of the main purpose of these messages. So these are super important. You're always going to want to include trial variable messages. Okay, let's go back here. Then at the end of the trial, you would send a trial offset message, and that can just say trial_result, all capitals, and then you can just put 0. It just needs some value afterwards, and you can just put 0. And that just marks the end of the trial, and it kind of complements the trial ID message that's sent at the beginning of the trial. So you can see that these messages they're not really that complicated. You know, it's just a matter of sending simple text messages that fit the formatting that's expected by Data Viewer. Let's talk briefly about message offset values. So in the example that I showed you here in Psychtoolbox, we're drawing to the screen, then we're sending a event marking message, and then we're sending the image load and interest area messages here. And in Psychtoolbox, because it uses, I guess what you can call like retrace locked drawing. So basically, like, this is where we draw the stimulus here, and Psychtoolbox won't release the code to execute the next line until the kind of millisecond when the screen is updated or when the command is sent to the screen to update. And so in this case, if we put the next line is a message to the EDF file, then that message will be time locked to the time when the command is sent to the monitor to update the screen. And if you're using a fast monitor, it'll basically be kind of synced up with the time of the drawing on the screen. And let me mention, these messages are sent super fast. You can actually send, if you wanted to verify this, you could send like several messages back to back to back. Like, see how we're sending several messages back to back to back here. When you do that, then if you check the timestamp in the data file, they'll most likely be within the same millisecond. So it's super fast. It takes much less than a millisecond to send these messages to the data file. So you don't have to worry about really like delays through this stuff. If you do send a lot of messages in a row, then like, let's say you sent like 20 messages in a row or something, then it would probably be a good idea to put like a 1 millisecond pause after every 5 messages, just to ensure that you're not like flooding the host PC out with too many messages, because they'll be sent super fast, and with some of the older eye trackers, you could potentially like overload the EDF file, and it might miss some messages, but that's probably not going to happen in the newer eye trackers. But just to be safe, you would probably want to put a 1 millisecond pause after every 5 messages. But anyways, in some programming environments, you can send messages in a way that's going to easily be synced to the time of that event. In other programming environments, that might not be possible. And so in those other programming environments, or just maybe because you're programming things and you want to do things in a certain way, you don't want to have these messages at the point in your experiment when the drawing occurs. You want to send the messages later, but you want to kind of like back timestamp them or forward timestamp them. So in some programming environments, you might send the message after the event occurs and then back stamp it in a way through the use of an offset value. And in some programming environments, you might know that some event is scheduled to occur X milliseconds in the future. And then in that case, you would forward timestamp it through an offset value. So what I'm trying to say here is basically when you send a message, if that message starts with a number and then a space after that number and then has the message text, then that number will be subtracted from the timestamp of the message in the EDF file when positioning that message in time in Data Viewer. So when Data Viewer imports the EDF file, whenever it encounters a message that starts with a number, then that number will automatically be considered to be an offset value, and it will subtract that time from the timestamp of the message when it's positioning the message in time in the Data Viewer interface. So the idea is, like, let's imagine you cannot, for some reason, send a message at the time of the experimental event. Then you just have to compare basically what time is it right now, what time was it when that experimental event happened. And in almost any programming environment, that's really easy to do. You just check current time, check the time of this past event, and then subtract those values, and that will give you an offset. And then you include that offset value as part of the message text. And in this case, this will move the message backwards in time 650 milliseconds. And then if you know some event is going to happen in the future, like let's say 650 milliseconds in the future, then you can say, what time is it right now? What time is the event scheduled to occur? take the difference of those and then send that numerical offset as the beginning of the message text, and then that will move the message forwards in time. And Experiment Builder does this actually automatically all the time. So let me just show you. I didn't point this out, but in our target position messages, Experiment Builder always knows the time of the upcoming scheduled screen retrace. And so it actually like writes the messages in advance of those screen retrace with a future kind of offset value here. And so this message, instead of being interpreted as occurring at 89551964, it would be interpreted as occurring as 89551969. Okay, so it moves it into the future for you automatically. So in the case where you cannot send a message at the exact time of the experimental event, you can always use the offset trick to just position it at the correct point in time. Okay, so that's all that we have to discuss. That's the kind of detailed overview of the Data Viewer integration messaging protocol. And thanks everybody for attending. And again, don't hesitate to let us know if you have any questions, and happy tracking.